University of Michigan. She was also an associate dean for graduate and professional education at the University of Michigan Engineering School. So welcome, Marianne.
So
we can get rid of that.
all right.
Okay, good. So now you can share your screen.
Just go ahead. Oh, I see. Share screen. Let's launch.
Good. That's good. And then I'm just going to get rid of this. Since it's in the way.
Okay, all set.
Oh, did I just?
Oh,
Oh,
I don't know. Or should we should stop share?
no, it's okay. Can you just tell me which one?
Maybe stop share and then oh, maybe here.
Okay. Is that good?
Great. That's good.
Okay.
Okay, well, thank you very much. Good morning, everyone. Can you all hear me in the back? Okay, is that great? Okay, good. Well, listen, thank you so much for that kind introduction and the invitation to be here today. It's really a pleasure. Thank you. It's really a pleasure to learn more about your department and meet all of the faculty and staff and students and see your beautiful campus. So it's really a pleasure to be here today. So what I'm hoping to do is talk to you about using optical diagnostic technologies for disease detection in the pancreas. See? That doesn't seem to be, okay. Let's see. How about, there we go. Okay, everyone can see. Pardon me? Okay. All right, thank you. Thanks. So let me just begin by describing some medical devices that are based on optical technologies for tissue diagnostics in clinical settings. And so of course, everyone is very familiar with the pulse oximeter. Any of the BME students in the audience or BME grads probably built a basic version of a device like this at some point during your education. And so the idea here is that near-infrared light is being passed through tissue. There's a light tissue interaction mechanism, and certain wavelengths of near-infrared light are absorbed differently by oxygenated versus deoxygenated hemoglobin. And so as the light passes through the tissue, it's detected on the other side. And we're using this phenomenon of near-infrared absorption to measure blood oxygen saturation. So this is an example of a device that we're all very familiar with in clinical use today. Another example I'm choosing is an imaging modality. So optical coherence tomography, or OCT, is used to create a three-dimensional image of the retinal structure. And so what happens in this device is let me see if I can use this. Here, can you see my mouse? Okay, so a patient rests their chin and forehead against the device here. In this case, there's a form of laser ranging going on. So light is shown through the front of the eye to the back. It penetrates through the back, and light is going to be backscattered when it encounters a difference of index of refraction in the tissue. And so what happens is this laser beam is raster scanned across the back of the eye, and a three-dimensional image of the retina is developed. And you see it here sort of coming off layer by layer. And so here the fundamental light tissue interaction mechanism is scattering or reflectance. So here's some examples of medical devices that are in clinical use today. And so more generally, we can use tissue optical sensing, spectroscopy, and imaging to probe local tissue biochemistry and morphology. And when we do that, we can use them in vivo for diagnostic applications. And there's a lot of potential advantages here. So of course, these are non-invasive technologies. And if we know something about how light propagates in tissue and how it interacts with tissues, we can be quantitative. We can develop mathematical models that I'll show you later, where we can extract quantitative information. The two examples I just showed you are label-free sensing. So only light is being shown onto tissue, but you can also use contrast agents if you think about fluorescent dyes to enhance contrast for different applications. The technologies can be portable or fairly small. They can work very quickly, essentially in real time, and be cost effective. And the smallest technologies can even be disposable. As I mentioned, we're measuring tissue biochemistry and morphology. So we're sensitive to tissue function and structure. And there's a wide variety of light tissue interaction mechanisms like optical absorption, scattering, fluorescence, Raman activity, and so forth that we can be sensitive to. And then finally, these techniques are compatible in a multimodal sense. So we can use multiple techniques together. They can be compatible with ultrasound or MRI and so forth. And we can use them in different modalities, point sensing, imaging, mapping, and so forth. So there's a lot of flexibility here. So studies in the pancreas are relatively few in number. So we began this project some years ago when a clinician, Dr. James Scheinman, who is the director of medical imaging at endoscopy at the University of Michigan Medical Center, came to us out of concern for his pancreatic cancer patients. And he explained to us about the clinical diagnostic need for pancreatic cancer patients. I'll explain that in a bit. The inaccessibility of the organ is really a challenge. And so in this image here, you can see the pancreas sort of buried in the middle of the abdomen. It's at the bottom of the stomach, so very hard to access. And when we began thinking about using optical diagnostic approaches to sense pancreatic disease, we realized that very few human studies had been published. So we were really starting from scratch when we began this project. And so what Dr. Scheinman described to us was that pancreatic cancer has a very low survival rate, and it hasn't changed in many decades. What I'm showing here is a plot of 5-year survival rates for many different forms of cancer and by decade. And so you can see that in general, the survival rates have been increasing, which is a good thing. But when we look at pancreatic cancer, these rates are very, very low. And it's just been very challenging to diagnose this disease, particularly at early stages of development, where it can be treated effectively and then increase those survival rates. So this was the clinical situation. So the project that I'll talk to you about is looking at whether we can improve the reliability of cancer diagnostics in the pancreas, particularly at earlier stages of development. So I'll talk first about the clinical challenge as it was described to us. I'll talk about a proposed solution. So we'll look at the rationale for using optical diagnostics, which really had not been explored previously. And then finally, I'll show you some preliminary results, pilot studies that we did in the clinic on human patients, and then more recently, steps toward the miniaturization of that technology. Okay. So when it comes to pancreatic cancer, the diagnostic methods that are available to clinicians are really unable to reliably detect it, particularly at early stages of the disease. We've already talked about how inaccessible the organ is. So techniques like ultrasound, CT, and MRI can detect masses, but usually when they're larger in size. If we want to detect it at earlier stages of development, we'd want to find smaller tumors, potentially less than 1 centimeter in size. The best methods for detection are endoscopic, and I'll show you how that works in a minute. Unfortunately, they have low sensitivity and specificity. So once again, another clinical challenge. So the best endoscopic method is known as EUS-FNA, endoscopic ultrasound guided fine needle aspiration. And if you're not familiar with that technique, an endoscope is introduced through the mouth and down the esophagus to the base of the stomach, and it's brought into proximity to the pancreas, okay? So there's an ultrasonic transducer at the tip of this endoscope. And so an ultrasound image, kind of depicted here, goes through the wall of the stomach and into the organ of the pancreas itself. The idea in this diagnostic technique is that a mass can be visualized under ultrasound, but they don't know what that mass is. It could be cancer. It could be inflammation, something known as chronic pancreatitis, which very often occurs. In fact, cancer very often occurs in the setting of chronic pancreatitis. So it's very difficult to sort out. So they can see that there's a mass, and I'll show you an image of that in a moment. And what they do is they introduce a needle. Okay, so the needle comes out through the end of the endoscope. Here, it goes through the stomach wall and into the pancreatic tissue itself, into the mass. And the needle is hollow. So there's an aspirate of the cells and tissues in that mass that's taken. This cell and tissue aspirate is smeared on a microscope slide, and right in the endoscopic procedure room, they look under a microscope to see if they can identify features consistent with cancer or inflammation or normal pancreas. Very challenging to read, challenging to take the specimens, challenging to read them in real time. Usually the needle can be passed a few times, sometimes as many as 7 times, but it's very difficult for the clinician to do this, and of course for the patient to tolerate it. And so in the field, there's what's known as a diagnostic dilemma, because cancer very often occurs in the setting of chronic pancreatitis or inflammation. The sensitivity of this technique is only about 54%. So that's not much better than flipping a coin. So that's really very, quite challenging and unacceptable. Even the negative predictive value is 72%, meaning you can have this procedure be declared negative, free of cancer, but really you're only 72% sure that that's the case. So this is really challenging for clinicians. And let me just show you the ultrasound image here. So I think you can see that there's sort of a darker gray area in the middle. That's the mass. And the endoscopist has to, bring the needle into proximity and then make a pass through that mass, take the aspirate, try to get the read, go back and do it again and again. So it's very challenging. Okay. So from the clinical perspective, patients often present with overlapping clinical symptoms that might be abdominal pain, weight loss, jaundice, and so forth. It could be cancer, or it could be chronic pancreatitis or inflammation. They're very commonly sent for diagnostic tests and diagnostic imaging, and may be referred to that procedure that I told you about, endoscopic ultrasound guided fine needle aspiration. This is going to make the definitive test. But of course, we saw the challenges associated with that technique. If there isn't a definitive diagnosis for cancer, a patient will not be referred to surgery. They will wait and typically come back later for more diagnostic testing. And so patients are kind of stuck in this loop here while they wait for that definitive test. When they are referred to surgery, it's a major surgical experience. It takes 7 or 8 hours or longer for this surgical procedure. We're going to go in during surgery and take measurements during this procedure. But I don't know if you can see it. It's covered up a little bit by the captions, but even patients who've been referred to surgery and have the procedure, in about 9% of cases, it was benign tissue only that was found. So it's hard to detect it. It's hard to make sure that you don't have it. And so the essential question that we were asking is, can we improve, can we develop another source of contrast? We know that ultrasound can detect a mass, but it can't tell us what that mass is. Is it cancer or is it inflammation? Would there be more contrast through optical diagnostics that would help us distinguish between cancer and inflammation? And so that's the question that we're going to ask. So let me explain the rationale behind why we thought there might be an opportunity here for optical diagnostics. I've already told you that through optical sensing, light tissue interactions, we can probe tissue biochemistry and morphology in situ. And so what I'm showing here are histopathology slides of normal pancreas, inflammation or chronic pancreatitis, and then cancer, pancreatic adenocarcinoma. And so if you look at these slides, what you'll notice is there's much more stromal collagen in the diseased tissue versus normal tissue. So it shows up with that sort of lighter pink color. Well, we can use an optical technique called fluorescence to detect collagen. actually. And this is without any labels, so it's a label-free technique. If you send the right wavelengths of light into tissue, the collagen in the tissue will fluoresce. And you can collect, using optical methods, that fluorescence signal and get a sense for how much collagen is there by essentially how much light you are collecting from fluorescence. And so the idea is that because of this increase in stromal collagen, we should be able to use fluorescence, this is endogenous label-free fluorescence, to distinguish diseased from normal tissue. So that was one piece of the rationale. The other thought we had is that if we look at cancer tissue, the cells here have enlarged nuclei, noticeably enlarged nuclei. We know that when light interacts with tissue, refractive index changes, the sizes and shapes of scatterers in the tissue will change the way the light interacts with tissue and scatters. So we can use a technique called reflectance spectroscopy, which sends light into tissue and measures the light that is scattered. And so reflectance spectroscopy should be able to tell us which tissues have larger cell nuclei in their interrogation volume as we deliver light to tissue. And all of this can happen very rapidly. We can send pulses of light. As you know, light travels very quickly. We can send pulses of light into tissue and detect these very rapidly. So potentially, if we could use fluorescence and reflectance with label-free sensing, no contrast agents, we could non-invasively detect the presence of chronic pancreatitis or cancer in the tissue. So that was the rationale. And there was a lot of research in other tissues, not the pancreas, because it's very difficult to reach that organ, but there was lots of diagnostic information in other kinds of tissues to suggest that this might be an approach. So what we did was we built a research tool. So this is a photograph here on the left of a clinical device. So this was our prototype. We had a broad spectrum lamp to illuminate tissue to collect reflectance signals. So the light was delivered to the tissue via fiber optic probe assembly. We had a laser of an appropriate wavelength to excite fluorescence from the tissue. This is endogenous fluorescence, again, delivered to the tissue via fiber optic probe. And then we had a detector fiber to collect the light. They were sort of illuminated sequentially to detect the light and then measure it spectrally. And I'll show you all of that data. And all of this was controlled, with interfaces with a laptop. And it was put onto a cart with wheels so that we could go into the surgical suite. What we realized we needed to do was we needed to take measurements on human pancreatic tissues that were matched to histopathology. So in other words, we're going to take the pancreatic tissue, bring the fiber optic probe into proximity, sort of touch the tissue, illuminate it very rapidly to collect fluorescence signals and reflectance signals, capture that data, and then immediately take a tissue biopsy right at the site of interrogation, so that for every optical measurement we have the gold standard as well. We have tissue histopathology. And so we received permission from the medical center to do exactly that. And so this is just a photograph of our prototype device. This is in the surgical suite. So these are patients undergoing the Whipple surgical procedure. This is a surgery that takes many hours, typically 7 hours or more. And so this is one of my former graduate students, Bill Lloyd, who's there with the device. And what will happen is the surgeons, we receive permission to take measurements on tissue that was recently excised. So they would go through the procedure, remove the pancreatic tissue, and then immediately give it to us so that we could take some measurements. And so I'm just going to show you a picture of that. So here's an example of the fiber optic probe being brought into proximity. You see that little glowing spot of light. We had typically a graduate student from biomedical engineering there taking the measurements, and there was a pathologist watching very carefully where that fiber touched the tissue, where the measurements were taken, and she would take a tissue biopsy. And all of that was coded and sent histology analysis. So for every optical measurement, we know if it's normal tissue, if it's cancerous tissue, inflammation, or something else. And so we had permission to do that for a number of patients, and this is what we saw. So this is something that my student at the time saw right away in the very first few patients that we investigated. And it's also what held up over hundreds of measurements on human pancreatic tissue. So let me describe what we're seeing here. So I have fluorescence spectra on the left-hand side and reflectance spectra on the right-hand side. Normal tissue is shown in blue. Pancreatitis or inflammation is shown as sort of this gold color, and then red is cancer in both of these. And so what we see here, this fluorescence spectrum is actually largely composed of collagen fluorescence in this region here, right around 400 nanometers. And what you'll see is there's more collagen in both the pancreatitis and the cancer relative to normal tissue. And this is averaged over hundreds of measurements. Okay, we also see a dip in the middle here. That's due to hemoglobin absorption from blood. This is freshly excised tissue. So we also have an absorber in the field of view, and I'll talk a little bit more about that later. On the reflectance side, we notice a large increase in reflectance signal here, right around 500 nanometers from the cancerous tissue, less so from the pancreatitis and the normal tissue. So these differences were very easily detectable and recognizable just by eye. And again, we saw that from the initial measurements and then over hundreds of measurements. So what we realized we could do is we could correct, let me just go back. We could correct for the amount of hemoglobin. So these dips and features here around 400 nanometers have to do with hemoglobin absorption. We could actually fit the data using mathematical models and correct for artifacts associated with hemoglobin absorption. And so let me show you how these models work. There's something that we developed over many years. Ultimately, we ended up with a photon tissue interaction model for fluorescence. So we started with what we call the canonical normal pancreas. We had hundreds of measurements of normal pancreatic tissue that we defined as our normal pancreas, normal pancreatic tissue. We were able to account for both scattering in the tissue from those cell nuclei, as well as collagen fibers. And so there's models for doing that. We could also correct for absorption from hemoglobin and then calibrate for the fiber optic properties. It actually matters how you deliver light and detect light. And so we were able then to produce a best fit model to the data that we collected. So let me show you what that looks like. So what I'm showing here on the left-hand side is data from chronic pancreatitis or inflammation. Right-hand side is cancer. And the data that we collected, what was measured, is shown in green. And the red dashed line are the model fits. So you see we're able to fit the reflectance data. We're able to collect from the reflectance data information on the scatterers in particular, the sizes of the scatterers. We're also able to account for hemoglobin absorption, and we can use that then to correct the fluorescence that we measured, where that had that hemoglobin absorption artifact. And so here again, the measured fluorescence down at the bottom is in green. The model fit, the intrinsic fluorescence, once we correct for it, is shown in blue, and then the red is a PTI model fit. And so then we're able to quantify the amount of collagen in the fluorescence signal without being subject to the artifacts associated with hemoglobin absorption. So all of that to say that we can take the data that we could very easily see by eye, see the differences, and actually quantify it and pull out quantitative parameters, numbers. And so let me show you what that looks like over dozens of measurements. So these models were able to look at the reflectance data and pull out a morphological parameter. So this is the relative increase in size of scatterers relative to canonical normal pancreas. And so you see from normal tissue here, chronic pancreatitis in the middle, and adenocarcinoma, there's a big difference in the size of the scatterers, which is what we know is true, of course, physically, but we're able to take these numbers. And then, remember, using that information, we can get quantitative information from fluorescence on the amount of extracellular collagen in the tissue that we know is going to change with disease. And so this biochemical contribution to the signal from collagen, again, for normal tissue, inflammation, and cancer. And of course, we see a big difference when it comes to the inflamed tissues and the cancerous tissues. So now for every measurement that we take, we have the site-matched histopathology, we have a model so we can fit the data, we can extract parameters. And so we can begin to develop a diagnostic type of tool. And so we had permission to study 18 patients. So we did that, going into the Whipple surgery each time, taking data on multiple sites, multiple measurements, dozens of measurements, and overall hundreds of sites. This was all done immediately after resection within just a few minutes. And we have the histopathology. So what goes into this, this scheme that we use to come up with diagnostic probabilities for this technique was all of the data that we had, reflectance and fluorescence. All of this spectra was analyzed in multiple ways. We used something called principal components analysis, the PTI, the model, mathematical model that I told you about, and then a combination of the two. We were able to get out these parameters, and then we brought in a biostatistician from the Cancer Center to help us because we wanted to compare these multiple data analysis methods, but we recognized that we had 18 patients, but measurements on over 100 tissue sites. So there's a possibility that we can have intra-patient correlations in the data. So they had a nice scheme using generalized estimating equations, GEE, to account for that potential correlation that might appear in the data. So we developed this rigorous statistical method to account for the fact that we had many more sites than patients. And at the end, we could come up with these diagnosis probabilities, the probability that a tissue measured was normal or inflamed or cancerous. And here's what the data looked like. All right, so this is being plotted here on this ternary plot. So let's see, along the left-hand side is the probability that a site is cancer, adenocarcinoma. The highest probability would be right here at the top of the triangle. Pancreatitis was here down at the left, and normal tissue is here on the right. And the color coding here, the blue dots are each of the normal pancreas tissue sites that we measured, about 40 sites. Green is pancreatitis or inflammation, and red is cancer. And so you see how these are sort of plotted out in this fashion. What we can do then is we can draw a line and say anything above this line in this area of this graphic, we're going to call cancer. Anything below, we're going to call benign, and so forth. And when we draw the line at this location, just to give you a sense of the sensitivity, specificity, positive and negative predictive values, we have about 90% sensitivity and specificity, and closer to 80% positive predictive value, 94% negative predictive value. If you remember before, the sensitivity that we had was something closer to 50% in the clinical procedure. So, and negative predictive value was more like 70%. So we felt that this was very promising. Obviously, the optical method could detect differences in the tissue very rapidly, compatible with minimally invasive technologies and ultimately endoscopy. Well, with any diagnostic, what you can do is you can take this threshold and you can move it. And you can get develop something called a receiver operating characteristic curve. This ROC curve is used in clinical diagnostics, and it's plotted here on this graph, showing the sensitivity on one axis and 1 minus the specificity on the other axis. Perfect sensitivity and specificity would be right over here, right at 1.0, okay? As we move that threshold around, we can create these ROC curves to give us a sense of how good our diagnostic is. And we're showing this for different types of parameters. So if we take all of the data, reflectance and fluorescence, we get this blue line. Reflectance only is shown in red, and fluorescence only is shown in green. And I should just mention on these types of curves, a 50-50 chance. Flipping a coin would be a diagnostic that was right here in this diagonal in the middle, okay? So we're doing much better than that. But what we also noticed from this data, and which I'll come back to a little bit later, is how much better the reflectance did than the fluorescence. And so that's something that was interesting to us. Again, this was the first investigation of its kind, and so this is going to be something that we can come back to later on. All right. I just want to mention a couple of points. As we were collecting data, there were a few sites that had a different diagnosis from histopathology. There's a tissue type called IPMN, intraductal papillary mucinous neoplasm. And it's not normal pancreas, it's not cancer, but it's considered a precursor to pancreatic cancer. So it's destined to become cancer, even though it is not yet cancer. We only had a few sites like this, okay? It was a limited study, but we found it very interesting. When we looked at the data that was collected from those sites, they recapitulated the features that we found from cancerous sites. So here's an example. Green is normal, adenocarcinoma is red, and as you can see, the IPMN really recapitulates the features that were found in cancer. So it's potentially a method to diagnose or detect precursors to cancer in the pancreas, which was very exciting. And similarly for fluorescence, which is shown here. So again, green is normal pancreas, cancer is shown in red, and blue is the IPMN, and it really recapitulated that. So we thought that was interesting. One last study we did was actually taking measurements in vivo. Everything I'd shown you was taken ex vivo. That's what we had permission to do. We went back to the hospital and sought permission to make these optical measurements inside of the patient during surgery, so the tissues were living. We could only do this on a few patients, but they did give us permission to do that. And so we developed a method using an angiocatheter. So during surgery, here we inserted an angiocatheter, and we used that to guide a fiber optic probe into place, very rapidly collected measurements. We removed the fiber optic probe. They continued with the surgery, resecting the pancreas and such as part of this Whipple surgery, and leaving the angiocatheter in place. So it was kind of a marker locating where that tissue site was. When that tissue was removed, we could reinsert the fiber optic probe to at least approximately the same location. And so in that way, we were able to have site matched in vivo and ex vivo. It's not exactly perfect, but that was the best that we could do. And then of course, pathology following from that. And so let me just show you what that data looks like. So here is, and we could only do this on a few patients, but here are the reflectance spectra and fluorescence spectra from cancers, both in vivo and ex vivo. And there's qualitative agreement. The differences are really coming from blood absorption, blood in the in vivo tissue versus ex vivo where it is draining away. Let's see here, this is adenocarcinoma in vivo, so cancer in vivo versus normal in vivo. And you see that it recapitulates that same feature and reflectance that we anticipate from cancer. So we are hopeful that this would work in the in vivo setting. We could only do that on a few patients. Okay, so just to summarize this section, I just want to emphasize this was a pilot study on a few patients. These were surgical patients and largely ex vivo data. But the diagnostic technology did work. It's possible to take these measurements. It's portable, rapid. It's entirely label-free. Potentially, it's compatible with endoscopy. These types of measurements, a fiber optic probe like that could be inserted through the endoscope, through the hollow needle, and very rapidly take measurements, potentially guiding biopsy during an endoscopic procedure, which could be helpful. And again, these were the first studies of their kind. And we believe that using tissue optical spectroscopy with quantitative modeling can provide metrics that could be useful for disease detection in the pancreas. Now, I just want to come back to this point about the portability of the technology. So remember that the tool that we developed was portable. It's on wheels. It can be wheeled in and out of surgery, but it's not exactly very small. And remember too, that most of the diagnostic power was in reflectance. So all of the tools that we had here to measure fluorescence, the lasers and so forth, we didn't really need in the end. Also, the reflectance full spectra, which is shown here, isn't necessarily needed either. If you think about it, if you just measured reflectance at 2 wavelengths, say around 470 nanometers and 630 nanometers, just by forming the ratio of the reflectance at those wavelengths, you could have similar diagnostic discrimination. That was the thought. Okay, so if we do that, then we're really simplifying the technology considerably. We don't need spectrometers, we don't need fluorescence, we don't need lasers. And just showing the example of what would come from that data. So if you were just to take the reflectance ratio at 2 wavelengths, 630 and 470, you get fine discrimination between cancers and inflammation in normal pancreas. So we went ahead and developed an optoelectronic microprobe. And I'll show you the details of that. So basically, we're replacing this technology here with just a tiny little sensor. It's an optoelectronic sensor that can fit through the hollow needle. Let me show you what that looks like. So this is a schematic of the probe. So this is a 19 gauge needle that's used in fine needle aspiration. Remember, it's a hollow needle. So we have here inside of what we're calling this viewing window, which is really the window that's used for the aspiration when they do that procedure. We're putting in this optoelectronic assembly. And so we're using micro LEDs at 2 different wavelengths, 470 and 630. So these are going to be our light sources that we can turn on. And we have a detector here in the middle. Okay, and the spacing here is designed to replicate what we were using in our fiber optic probe assembly. So it's about, I think, about 660 microns center to center spacing. So we're going to see now if we can construct this and take measurements and actually recapitulate what we did with that larger sort of research instrument. And so these are just some photographs of the technology itself. So over here on the right-hand side is the sensor module that we fabricated. Here's a close-up of the module inside of the 19 gauge needle, the hollow needle. And here's a picture with those micro LEDs lighting up, obviously very bright. So we can turn on the red one, take a measurement very quickly, turn it off, turn on the blue one, take a measurement very quickly. And the idea is going to be to see if we can use this technology to replace the other technology. What I'm showing here on the left-hand side is a schematic coming from a code. Remember, all of the measurements that we took on pancreatic tissues, we extracted optical properties from those measurements. So it wasn't previously known what the optical properties of normal pancreas, inflammation, cancer, and so forth were, but we had that information. So we could put this into a code for photon migration, where we would send photons into pancreatic tissue and then see where they went, essentially. And so what's shown here in blue are paths of photons as they are launched from the 460 LED. We're looking at which ones ended up at the detector, i.e. which ones did we measure. And so if you run that code multiple times, you come up with a path roughly like this. So you see we're penetrating about 500 microns into tissue, roughly interrogating that region. That's very similar to what we were doing with the fiber optic probes. Similarly, for the red LED, I'm showing here the paths of photons. If you launch, millions and millions of photons, the paths of those that reached the photodetector in the middle. And so this penetrates a little bit deeper as red light would. And again, something around 0.5 millimeter. So as you light up these, you get sort of a glow ball that's very consistent in terms of the interrogation volume that we're detecting. That's very consistent with what was measured under the fiber optic probe. So that was the idea. And we went ahead and calibrated it and developed some tissue phantoms and took measurements, and we compared the large instrument with the optoelectronic microprobe assembly. So this is reflectance data here on these phantoms. So obviously the full spectrum is coming from the larger research instrument that we have. And then these individual data points at these 2 wavelengths is coming from the optoelectronic microprobe assembly. So we have, the instrument is working as we would anticipate, and we have a good correlation between those ratios that we're going to calculate at the end of the day. So this is just a verification of the instrument. So once again, we got permission from the hospital to take measurements on some patients, in this case, 4 patients. And so here's an example of the probe being brought into contact with the tissue and the measurements being taken. And down here at the bottom, you can see the data from the different patients, patients 1, 2, 3, and 4. So the pancreatic cancers are shown here in red, the inflamed or chronic pancreatitis tissue in blue, and then the normal tissues in black. And you can see that we're plotting this against the reflectance ratio, and you can see how they group based on their histopathology. And so that's what's shown here at the right. So this is the reflectance ratio for these different types of tissue, normal tissue, inflammation, and cancer. Very similar to what we saw with the full-fledged instrument, but now with the little microprobe. Okay, and so I just wanted to tell you that that's our latest work on this technology. We've certainly really capitalized on what we learned from the research instrument and now really doubled down on the portability of that instrument. And so with that, I'd like to acknowledge our team. So at the University of Michigan, we play football, and there was a very famous football coach who had a saying, it's the team, the team, the team. And I really believe this here. We had an incredible group of very dedicated faculty, students, postdocs over the years who contributed to this project. And so I want to acknowledge Jim Scheinman. He was the one who initially brought this project to us, and we worked with him for many, many years. Diane Simeone from surgery, Barb McKenna from pathology, and all of the other surgeons and pathologists. Jeremy Taylor helped us tremendously with biostatistics for this data set. And then there were a number of postdocs and graduate students from applied physics, biomedical engineering, and biostatistics as well. And I'm very grateful to our funding sources. We started out with internal funding from the University of Michigan. The National Pancreas Foundation funded us as well. The Coulter Foundation for Translational Research supported our project, and finally, the National Institutes of Health. So I'm very grateful to all of them, and I thank you all for your attention.
Thank you so much. So if we can stop sharing here, and magically somebody going to control this window. So thanks so much for fantastic. I think it's really very, very fantastic, very impressive in terms of basic science actually able to translate into bedside. It's really the bench work to bedside. That's fantastic. So before I hand it to Professor Lisa Olson, and please follow us on social media to receive latest update. You can see our social media information on the slides. You can always access the registration information for our BME seminar, our website at bme.columbia.edu. Now I pass it to Lisa to handling questions.
Okay, thank you very much.
So, okay, so far, no questions from the, oh, we have one from the live virtual audience. So let's start with the students.
I see one.
Oh, great.
Oh.
So maybe in the back first.
Yeah, I was gonna ask, if the sampling depth is on the order of 1 millimeter, how common is it to have pancreatic?
Stand, we can't hear you.
Sure. So I was gonna say, if your sampling depth is like on the order of 1 millimeter, how common is it that you have pancreatic tumors that are not located kind of proximal enough to where the endoscope could go, that you just can't detect them?
Yeah, no, that's an excellent question. So should I repeat the question too? It has to do with the sampling depth. And as you are, for example, especially if you are inserting a needle into a large mass, right? How do you know, how likely are you to be then encountering a tumor, especially if it's embedded in inflamed tissue, et cetera? That is really an excellent question. And the idea behind this was to see, first of all, was there any contrast at all from the optical technique? And that's why we started out very broadly measuring every site. In fact, I guess I should tell an anecdote. The very first patient we measured only a few tissue sites. And it's because the pathologist who was with us in the room said, measure here, this is going to be cancer. Measure here, this is going to be normal. Well, when the pathology came back and we looked at the data, the pathologist had no idea where to measure. You just cannot tell, you just cannot see. So we learned to take multiple measurements because we really did not know where this was going to happen. The great thing about the optical technique is that it works very rapidly. So if you imagine incorporating this into an endoscope, you could use it to guide biopsy, where they take their aspirate, for example, or if it's accurate enough, actually render a diagnosis. So the concept was, and the reason to have multiple modules on that optoelectronic probe, I showed you one with 2, for example, in the schematic. The idea was that as the needle is inserted, measurements could be taken very rapidly. So actually you could acquire this data very quickly as the needle is inserted, hopefully, ideally, running into one of those sites that was cancerous or possibly even a precursor to cancer. So that would sort of highlight the best place to take the aspirate, or if it's accurate enough, actually render a diagnosis. But yeah, for most of this work, we were operating completely in the dark. And so we learned to take multiple measurements, given the limited patient set that we could acquire because of surgery and those types of constraints. But yeah, that's a really good question. Thank you.
My question might actually be along the same line with the question I asked before, but because of the fact that the needle is open, and because you're constantly in tissues, I guess as you're at the measurement, I guess it makes sense. So were you doing this? I guess maybe this is for clarification. Were you doing this on just the tumor tissue itself or on the pancreas?
Okay, so the data that was shown was on tissues that were typically, with rare exception, ex vivo. So the tissue was the organ was cut, the tissue was exposed, and then we would take measurements by bringing the probe into proximity, into gentle contact with the tissue to take those. In an endoscopic setting, what we envision is that there could be a protective sheath actually on top that's then retracted. That's very common in the endoscopic procedures, that they can manipulate those tools endoscopically at the distal end. But that's something that we didn't do because this was mostly ex vivo. Aside from the angiocatheter, which is where we had a fiber optic probe that went in, which was a little bit different. Yeah, it's a good question.
Okay, so let's see. I think we did have one up here. So Jet Lem asks, would a multi-photon technique be a possible choice to avoid the blood absorption and increase penetration depth?
Yeah, no, so the first question is actually, so using other types of light tissue interaction mechanisms, I think it's very possible. I think relatively little has been done in the pancreas. And so these are, this is a field that the clinical need is great. I think the opportunity is also great for biophotonic and other methods. So I think that's also a possibility. That is not something that we have done. I can read the next question as well. Okay, so the next question is, do you think it would be possible to get interpretive data through the gut wall, or is it necessary to get right to the mass with a needle biopsy? So for optical techniques, we are going to be limited to penetration depth. And so we really do need to be in very close proximity, essentially inside of the mass. And that was the vision. Yeah, so ultrasound could be used to guide to see where the mass is, but then possibly optical diagnostics, they're using those sources of contrast could be used to render a more definitive diagnosis.
Let's see if any more student questions. Oh, well, not a student, but professor.
Are you concerned about like you're shining light, right? Can you stand up? Sorry. Make it better. Hey, are you concerned at all about like obviously you're shining light directly at a tumor and like
Right.
you have to be worried about tissue decay?
Yeah.
You have to evaluate that at all?
Yeah, thank you. That's a really good question. So, we're not, when we do optical diagnostics, we're not putting down enough energy, enough light to actually damage the tissue, right? So we're trying to not damage the tissue actually, but to measure it with as little light as possible. So there's been a lot of work in the field designed to actually develop thresholds where you want to stay below that. And yes, so we're always sensitive to that. But that's a very good question, because if you change the sample that you're measuring, the tissue that you're measuring because of heating and other effects, then you could be changing the output. But we tried to use relatively little light. Yeah. That's a good question. And then I think there was another question before then, but it disappeared. Sorry. So does light scattering depth change for different wavelengths or different tissue properties, such as the nuclear size? So, light scattering is affected by the wavelength of light that you use, but usually not so dramatically. There's usually a lesser effect. Light scattering is going to be very sensitive to changes of index of refraction in tissue. So typically the size of scatterers, the shape of scatterers, and the density of those scatterers in the tissue. And we think that's why there is such a diagnostic yield when it comes to these cancerous tumors, where you have this enhanced nuclear size, large nuclear size, very clearly different than the surrounding normal pancreas. And that's something that's very well established in the field, hadn't really been measured as much in pancreatic tissues, human pancreatic tissues. So thank you.
Following up on the previous questions, if we're doing the microprobe measurements in vivo when it's in the patient, we inevitably still have to punch a bunch of holes through the endoscopy.
Yeah, that's a great question. So, and again, we haven't done this in the patient during endoscopy. That would be, you know, the ideal setting ultimately for this during the endoscopic diagnostic screening procedure. The challenge, of course, there is that we needed a gold standard for histopathology. So we had to have access to the tissue to take a biopsy. But the ultimate goal is that, and the thought is that, again, by taking rapid optical measurements, we might be able to sort of locate the area that's most likely suspicious. And then we could take a biopsy there. But it is true. And for some patients, my understanding is that they can tolerate a certain number of passes of the needle through the mass, sometimes as many as 6 or 7, but it's really challenging for the patient to tolerate that. So we're trying to have a less invasive, what is inherently an invasive procedure, be as less invasive as possible. So ideally, it would be nice if we could have fewer passes, but that is a challenge.
You only get 1 point with every pass.
So for the aspiration, when they make a pass, they take an aspirate. So that's how that works. For the optical measurement, because these measurements can be done so quickly, ideally, as the needle is inserted through the mass, we could take measurements along. So we're kind of doing a almost like a spectroscopic type of mapping, you know, in the broad sense, not really imaging, but mapping. That was the concept behind that. And so especially in the optoelectronic microprobe assembly, those assemblies could be very small. And so there could be multiple assemblies. And so this could be done very rapidly. That was the concept. Thank you, though. That's a great question.
So maybe we'll thank our speaker now and
Thank you. Thank you.
See you later.